Now we're into the nervous system. This is going to be 3 chapters because we're going to look at the basic structure and function of the neuron and the nervous system itself. Then we're going to get into the divisions of the nervous system, and then finally we're going to get into the senses. All right, so general characteristics of the nervous system. This is basically a master regulatory system. We're going to talk about the brain, and I mean that's our control. We're going to send and receive information, sensory input. We're going to integrate and process this, make decision about what goes on, and then we're going to basically decide whether we got to do something about it. And if we decide we got to do something about it, and we got to move something, or we're going to change something, there's going to be motor output, and that's how we maintain homeostasis. And also we know our brain acts as, where our thought processes, our learning, our memory, that sort of thing. The main cells of the nervous system are the neurons and the neuroglia. The neurons are the cells that conduct electrical impulses. We're going to talk about this. It's neurotransmitters. Didn't we just go over this when we talked about muscle contraction? So again, I said you need to really kind of understand that. We'll go into that in a little more detail. But this allows us to very quickly respond to changes or stimuli. You stick your finger in a flame, you want to very quickly get it out of there. The neuroglia is a protective tissue. It's going to nourish those neurons. It's going to insulate it. It doesn't conduct any electrical impulses, but it's a supportive tissue for those lovely little neurons. So the general flow of information is you have a sensory receptor giving you information. There's sensory input. It goes to the brain and the spinal cord, where it is looked at and decisions are made as to, do we need to react to it, do we not react to it, and if we have to react to it, then that information is sent out to the skeletal system or whatever it may be to have an action. So when we look at this, we have a central nervous system and a peripheral nervous system. The central nervous system, the brain and the spinal cord. The peripheral nervous system connects the central nervous system to all the other body parts. This is the cranial nerves and the spinal nerves. And we have 2 subdivisions. This is basically sensory and motor, the afferent and the efferent. All right. So the sensory division, basically, this is the sensory receptors that are going to sense something, detect changes, all right, and that information basically is going to be converted into impulses, and they're going to be conducted along those peripheral nerves to the central nervous system for integration and decision. And then the motor division, those neurons that transmit the impulses from the central nervous system to affect a particular motor function. So there are 2 subdivisions of the motor division, the somatic and autonomic. The somatic is to the skeletal muscles, the autonomic to the involuntary commands to the viscera, to the rest of the organs. So somatic is voluntary commands, autonomic involuntary. So if we take a look at it, these are our divisions. This is the central nervous system, and this is the peripheral nervous system. So we start looking at the cells. The neurons, they can run very, very, very long, several feet in fact. So they're going to vary in size and shape. They will differ in length and number and size of the axons and the size of the dendrites and whatever's going on, depending upon where it is. They're all going to share these features called the soma, or the cell body. And that cell body is going to basically look like a regular cell. You're going to have a nucleus, you're going to have a cytoplasm, you're going to have organelles, you're going to have Golgi bodies, you're going to have mitochondria, just like you were looking at a cell. There are going to be some chromatophilic substances called Nissl bodies. We'll talk about that. But the structure of that cell body basically the same, just like a regular cell. Then you have your dendrites and your axons. Dendrites are receptive surfaces. You can have a lot. Axon's going to transmit it. So the information coming into the cell body is coming in through the dendrites, and then it is going out through the axon. The axon is transmitting that impulse and releasing those neurotransmitters to another neuron or to an effector, a muscle cell, whatever, down the way. You can have one neuron may only have one axon. It depends on where you are. So when we look at the axon, we're going to be looking at this neuron in the neuron model in the lab. I'll be showing that to you. But you have the structure of the axon. You have the axon, you have the cell, and the cell body, and then you move into the axon. And that first portion is called the Axon Hillock. The hillock is a cone-shaped area where the axon is actually coming out of. And the terminal is the endings. Once you get down at the end of the axon, it terminates. You are going to have Schwann cells. This is the neuroglia of the peripheral nervous system that's going to wrap around some of these axons. The Schwann cell makes myelin. Okay, and myelin is a mixture of fat and protein, and it's wrapped in a sheath called the myelin sheath that coats some of these axons, and it acts as an insulator. It's like wrapping that black electrical tape around a wire, and that's what it does. It insulates these electrical impulses so they just don't go off anywhere. You have gaps in this sheath. We'll talk about it. It allows you to do what's called saltatory conduction, but you can basically hop, skip, and jump down the axon, making it much quicker, and use the gaps between these Schwann cells, and these are called nodes of Ranvier. So you can see there's our Axon Hillock, there's our cell itself, and you can see that myelin sheath. There is your Schwann cell, and these are the little nodes of Ranvier. So instead of going like this, you can go bump, 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 and it allows you to transmit a lot further. And you can see the axon terminal with the synaptic knob at the end. All right, so you have myelinated and unmyelinated axons. Not all the axons are myelinated. And as I mentioned, they are produced by basically you have a series of Schwann cells, and that Schwann cell is going to produce, and it lines up. In the central nervous system, you do have some myelinated axons, and it is produced by the oligodendrocytes. If you have a group of myelinated axons in the central nervous system, and it comprises what we call white matter. You have gray matter and you have white matter, and this is the matter that's, and we'll look at it, it's more internal in the brain. The unmyelinated axons are encased by the Schwann cell cytoplasm, but there's no coating of myelin around them. And the group of the unmyelinated axons in the central nervous surface make up what we call the gray matter. So that's that outer portion that we think of when we talk about the gray matter of the brain. Okay. All right, so you can see the difference between, here's the myelin sheath. So this is a myelinated axon, and this is an unmyelinated axon. Now we classify neurons by their structure, and it's just how many processes they have. A multipolar neuron is going to have a lot of processes extending from the cell body. So you've got many, many dendrites and one axon. This is most of the neurons. Most of the neurons in the central nervous system, some of them in the autonomic nervous system, but yeah, a large percentage of the neurons are this. Then you have the bipolar neurons. They have two processes from the cell body. So you got one dendrite, one axon. Not that common. This is your senses. And then you have the unipolar, or sometimes called pseudo-unipolar, but you have one process extends from the cell body, so you have two branches that function as one axon. And you may find these in the peripheral nervous system. So you can take a look and see what these look like. You can see this is, lots of dendrites coming in, one axon, one dendrite, one axon, and then got an axon kind of sticking out. All right, so we can classify the neurons by their function. Are they sensory? Are they motor? Or do they link? So if sensory, afferent, these are, again, carrying the impulses from these receptors from the periphery to the central nervous system to be processed. So at their distal ends, they're going to have these receptors to detect some kind of change. Most of these are going to be unipolar, just because, it makes for that function. The interneurons, these are going to link these neurons. They're going to be the relay stations, if you will. They're going to relay information from one part of the central nervous system to another. So these are going to be the multipolars. Your motor neurons, your efferent, are going to carry the impulses from the central nervous system to either a gland or a muscle or something to take that, to do that effect. In the somatic nervous system, they're going to control the skeletal muscles, and the autonomic nervous system is going to control the smooth muscle and the cardiac muscle and some of the glands. And you can see the different classes, what they look like, and how many we've got. So, yeah, got a lot of sensory neurons. We've got a lot of interneurons, and motors, not so much, because we're not always moving something. All right. So then we're going to look at neuroglia. And the general function of neuroglia, as I said, is to provide support. All right. It's going to basically produce growth factors. You're going to have growth factors that nourish the neurons and remove any excess ions or neurotransmitters. And again, it's just really supportive. And you've got a few different cells. The astrocytes, they literally do kind of look like stars. They're going to connect the neurons to the blood vessels that allow for the exchange and growth factors. They're going to regulate ion concentrations. They're going to form part of what's called the blood-brain barrier. This block between the brain and the blood. The barrier is, it makes it difficult for certain things to get into the brain. We want our brain to be protective, and there are times when we get stuff in the rest of the body. We don't want it in the brain. So this is kind of protective, but this also has a problem. If you've got an infection in the brain, it makes it a little bit harder to treat, because a lot of these treatments don't make it through very well. Then you have your oligodendrocytes. Again, the myelinated central nervous system axons. This is going to provide support. You have microglia. These are phagocytic cells. These are going to pick up debris. Then you have the epithelial cells. These are going to line the spinal cord and the ventricles. They cover what's called the choroid plexus. These are going to, what they do is regulate the spinal fluid. All right, because we've got circulating spinal fluid. So you can take a look at what these cells look like. Again, the Schwann cells are going to produce the myelin sheath. And this allows us, again, to speed up that impulse of that nerve transmission. This is a sheath that's going to cover it. It's not going to, it's again like this black electrical tape, so that you don't have these impulses that just move off and go off somewhere. It keeps that nerve impulse within that nerve, so you're protected. Usually with these, mature neurons do not divide, and if the cell body is injured, the neuron usually dies. Now, you can get some regeneration. In the peripheral nervous system, if the peripheral axon is injured, it may regenerate. And there is a process for this. There are sometimes, for example, in cardiac surgery where you cut a nerve, and you can't, that will regenerate. It may take months. A lot of times it may take 5-6 months to regenerate, but it may be able to regenerate. Central nervous system, those axons lack the neurolemma to act as a sheath, and the oligodendrocytes do not proliferate, and regeneration is unlikely. All right, so let's look at this membrane potential. Basically, the cell membrane is electrically charged, or what we call polarized. And basically, if you have a charge difference in an area, it is called polarity. The general rule is the inside of the membrane, the inside of the cell, is more negatively charged than the outside. So it's more negatively charged than the outside. The outside is usually more positively charged because of the ions. So you have an unequal distribution of ions on the inside and the outside of the membrane. And basically what's going to happen is we're going to have to move some of these charges. But that's important in the conduction of this. And these cells can rapidly change the charge. And that ability to change the charge is what allows these cells to work. So what we're looking at is what we call a membrane potential, or the change inside the cell. The potential to transport charges across the membrane. Okay, the potential is there, but you got to have something kind of tell it to start. We'll talk about that. So you have a resting membrane potential. The charge inside the cell when it's not active is about minus 70 millivolts. And the potential results from this unequal ion distribution. You have potassium ions and sodium ions. And like I said, it's more negatively charged in the interior. All right. Sodium ions are in higher concentration on the outside of the cell, and potassium ions are in higher concentration on the inside of the cell. All right. And basically it's slightly negative inside the cell because the large number of negatively charged proteins and ions that are in the cell. It's the proteins that are really adding to this. So you have a gradient of sodium and potassium. So on the outside of the cell, you have more sodium than you have on the inside of the cell. Potassium, you have more potassium inside the cell than you have outside the cell. What you have are channels that allow the movement of these ions to go back and forth. And you have chemicals and electrical charges that affect the opening and closing of these charges. So being able to have an active neuron depends upon these changes across this membrane potential. So, excuse me, inside the membrane you can see, here's your plasma membrane. You have positive charges on this side, you have negative charges on this side more, okay? So it's more negatively charged on the inside of the cell than it is on the outside of the cell. The outside of the cell has more positive charges, okay? And the sodium potassium follow the rules of diffusion that we talked about earlier in the course. They move from an area of higher concentration to an area of lower concentration across this membrane. All right, the membranes are more permeable to potassium than it is to sodium. You have a sodium potassium pump that maintains that balance. When you're in resting potential, when it's disturbed, you're going to pump 3 sodium ions out of the cell and 2 potassiums into the cell. Okay, and basically you're going to use the energy of ATP to actively transport this, because this is in opposite directions of that concentration. All right. So the action potential is a sequence of events involving the changes of this potential, first to positive, then to negative, then back to the resting potential. Okay, so you can just see here's the channel closed, the protein is closed, and here the channel is open. This is just a channel through your phospholipid bilayer. All right, so let's look at this pump. All right, so again, we're positively charged over here. You have a lot of sodium over here, more sodium over here than you have sodium here. You have more potassium here than you have over there. The sodium-potassium exchange pump ejects 3 sodium for every potassium that we move, okay? And this does maintain a stable resting potential, but you're going against the concentration gradient, so you have to use ATP. So you can go through that entire event and see what happens, but basically that membrane maintains a resting potential by diffusion of these down their concentration gradient. And when the neuron receives a stimuli, the sodium channels in the zone trigger, the sodium ions diffuse inward, which does what we call depolarize the membrane. The potassium channels open and the potassium moves out, which is called repolarization. And you have an action potential that causes an electrical current. Okay? And again, this is real important because you're going to get this again when you get to the heart. You're going to hear this again, so you should be able to get through this. But basically, you have a refractory period. This is the portion of the axon that is conducting. The axon is not able to respond. You are in refractory. You are not able to respond to another threshold stimulus. All right, you have an absolute and a relative. And basically, the absolute is when the threshold cannot generate, you just cannot generate another action potential, and the sodium channels are unresponsive. The relative refractory period is when you have a high intensity stimulus that can generate another action potential. It's a lot of stimuli hitting you. The repolarization is not complete, and you can move it. All right, so the speed of the impulse varies with whether you have a myelinated axon or unmyelinated or non-myelinated. Myelinated, again, we said is an electrical insulator. The ions can cross the membrane only through the gaps in the myelin sheath, the nodes of Ranvier. The myelinated axons transmit the impulses through what we call saltatory conduction, which allows these potentials to jump from node to node to node. It is a lot faster. All right. So if you sit there and look at a myelinated axon where you got to move it all the way down, it's 120 per second versus different. Okay. All right, so you can see this difference. So you can see this action potential, and it just begins to change, and this is the change in that potential as it what we call propagates down that nerve, down that axon. Okay. All right, and here's saltatory. You can see we can now jump. All right, and so that is it, and we're going to now go look at the different types of different sorts of the nervous system. We're going to look at the autonomic nervous system. We're going to look at some other things in the nervous system, and then finally we will close out with the senses. Okay.